Assalamualaikum and hi everyone. My name is Azian Azamimi Abdullah from University of Malaysia Perlis. Today I would like to present my research project. The title is Brain Tumor Radiogenomic Classification Using Deep Learning Algorithms. First, I will explain about brain tumor. Brain tumor is a type of brain cancer. It is 10th leading cause of death in the world. It is estimated about 18,600 adults that will die from primary brain tumor. For the patients aged 15 years old and below, the survival rate is around more than 75%. And for the patient which is in between 15 to 39 years old, the survival rate has decreased slightly, about 72%. And for the patient that 40 years old and above, the survival rate decreased rapidly, about only 21%. How to diagnose the brain tumor? The most well-known and widely used is the magnetic resonance imaging, or the MRI. MRI is a non-invasive method. It gives more detailed images compared to other imaging methods. The second widely used, it is the computed tomography or CT scan. It gives more information and it gives slices of images. And another medical imaging method is the positron emission tomography, or the PET scan. It requires the radioactive chemical. The scanner will produce image after they detect the chemical. And another method is a surgery. Surgery is an invasive method. It requires tissue sample to diagnose the brain tumor. For this project, we use the multi-parametric MRI scan, or known as MP-MRI. So the images has four modalities. The first one is the T1 weighted pre-contrast, T1W. It is dark CSF, low signal intensity. And the second modality is the T1 weighted post-contrast, T1GD. The bright signal in the blood vessels due to the gadolinium. And the third modality is the T2 weighted, or T2W. It has the bright CSF and high signal intensity. And the fourth modality is the fluid attenuated inversion recovery, or FLAIR. It gives the dark CSF and the long TE and TR times. Brain tumor can produce a specific biochemical product known as 06-methylguanine DNA methyltransferase, or known as MGMT promoter methylation. It is a type of cell's DNA. So if the brain tumor patient produces this specific biomarker, the MGMT promoter methylation, it will cause the chemotherapy drugs less effective. Problem statements. Chemotherapy drugs used to treat the cancer. However, the chemotherapy drugs can cause damage to tumor cells. And if the brain tumor patient produce the more MGMT, it will cause the chemotherapy drugs less effective. So the patient need to go for surgery. Surgery is the first treatment of brain tumor. It is require a tissue sample to be extracted, and the subsequent surgery may be necessary. This is the objective of our project. The first objective is to implement the data visualization techniques and exploratory data analysis, or EDA, on the brain tumor radiogenomic data to get more information about the brain tumor. And the second objective is to apply the image pre-processing and to implement the deep learning algorithms to predict the MGMT value of the genetic subtype of glioblastoma through the multi-parametric magnetic resonance imaging. So the scope of this project, we collect the data from the BRADS datasets. The dataset collection of brain tumor is obtained from the brain tumor segmentation. The BRADS will be implemented. And the second scope is to implement the EDA and the data visualization. The trained dataset will be explored and visualized for better understanding. Then the dataset of the MRI image will undergo the image preprocessing, and then we will determine the suitable deep learning algorithm for the prediction of the MGMT value. And for the whole project, we use the Python as the main programming language used for this study. For the literature review, based on the previous papers, for example, the first paper published in 2020, the title is Radiogenomics of Lower Grade Gliomas, the machine learning based MRI texture analysis for predicting the correlation status. So for this paper, they use the traditional machine learning. For example, the adaptive boosting, the k-nearest neighbors, Bayes neural network, random forest, stochastic gradient descent, and support vector machine. So the result is the machine learning based texture analysis of conventional T2W and contrast enhanced T1W MRI is a promising technique for predict the status of the brain tumor. And the more than 4/5 of the LGGs can be correctly classified as on unseen dataset. So there are a lot of papers. I'm not going to discuss in detail for the all papers. However, I will discuss on the research gap. So we found that most of the studies use the classical machine learning algorithms such as support vector machine, Neff Bayes, and also decision tree to predict the MGMT value. So for this project, we will implement the deep learning as a prediction model for the MGMT value of the brain cancer. And the MRI frequently used to diagnose the pathology of brain tumor, and the MRI data contain the phenotype features of the brain tumor. Those MRI as a source of information that may be utilized to predict the genomic of brain tumor patient. So this is the flow chart of our project. First is the data acquisition. We obtain the data from the BRAX MP-MRI scan, and after that, we will implement the exploratory data analysis and also the data visualization. So this is the first objective. And then we will implement the image pre-processing method for the brain tumor images. Then we apply the deep learning models. So for this project, we use the ResNet-50 and also the convolutional neural network to predict the MGMT value. And we evaluate the performance evolution by using accuracy and loss. This is the second objective of our project. And finally, we predict the value of the MGMT promoter methylation. So the methodology, first the data acquisition from the BRAX dataset, and after that we will go for exploratory data analysis and also the data visualization to understand more about the data, and then image pre-processing, and then we implement the two deep learning algorithms, which are ResNet-50 and also the convolutional neural network, and finally we evaluate the performance by using accuracy and loss. So for the data acquisition, there are two types of files, the training dataset and also the testing dataset. So each of the patient will contain the MRI scan taken from various different techniques in DICOM format. So there are 4 modalities: T1W, T2W, T1GD, and FLAIR, which I have been explained previously. So the testing dataset is another folder, so it also contains 4 modalities. So another file, also the CSV file. So the provided CSV file contain the label for whether the patient in the training data has the biomarker or not. It means it gives the value of MGMT either 0 or 1. So from the exploratory data analysis, we found that about 278 patients have the 0 MGMT value, and 307 patients have a value of 1 for the MGMT value. So it is about 52.5% of the patients have the MGMT promoter methylation in their brain tumor. So this is the biomarker for the brain tumor. And another 47.5% of the patients do not have the MGMT promoter methylation in their brain tumor. And after that, we implement the image preprocessing. We use the interpolation method. It is an upsample or downsample based on the original size and ratio to the target size of all scans to a standard size before being inserted and used into the deep learning models. So this is a 3-dimensional images, the X, Y, Z dimension. So we implement 2 deep learning models. The first one is the ResNet-50. So this is the architecture of the ResNet-50. And this is the architecture of the convolutional neural network. So it contains of convolutional layer, the rectified linear unit layer, the pooling layer, and finally the output prediction value. So our project is to input the image, and finally we can predict whether the image contain methylated or unmethylated. Methylated means this image has MGMT promoter methylation, and unmethylated means 0. That means this image do not have the MGMT promoter methylation. So results and discussion. So this is the graph for training data accuracy and loss. For the left side, it is the results of ResNet-50. The accuracy that we obtain is 0.8604, that means 86%, and the loss is 0.2410, that means 24%. And the right side is the results of the convolutional network or CNN. The accuracy that we obtain is 0.8521 and the loss is 0.2523. So it seems that the ResNet-50 give a higher accuracy compared to the CNN. And we also use the two models, the ResNet-50 and also the CNN, to predict the value of the MGMT promoter methylation using the testing dataset. So for example, for the patient number 1, the patient ID 00001, ResNet-50 give the prediction value of 0.9. However, for the CNN give the prediction 1. So it means that this patient number 1 has the high value of the MGMT. So the high value of the MGMT, it means that the chemotherapy is ineffective. So the doctor can suggest the patient to undergo the surgery to remove the tumor. So it is no point for this patient to go for chemotherapy drugs. Okay, so for example, for the patient number 6, the patient ID 00434, the ResNet-50 give the prediction value of the MGMT 0.3 and the CNN also give the same prediction value 0.3. It means that patient number 6 has the lower value of MGMT methylation promoter. So the low value of the MGMT, the chemotherapy is effective, so that the patient can undergo the chemotherapy treatment to remove the tumor. So this patient not required to go for surgery to remove the tumor. So it can just proceed with the chemotherapy treatment. Okay, so this is the whole testing data set. The ResNet-50 give all the prediction value for the whole testing data set. Okay, so 2 of the patient has the lowest value of 0.3 and the highest value 4 patient has the highest value of 1. It means that this patient has methylated, has the MGMT promoter based on the MRI scan. And this is the prediction results for the CNN. So only 1 patient has the low value 0.2 and the highest value of 5 patients. So conclusion. So we have 2 objectives. The first objective is to implement the data visualization and exploratory data analysis EDA on the breast data set. So from our project, from the results, the sample of brain tumor scan, the MRI scan images and their data has been visualized for better understanding. The second objective is to apply the image processing of the brain tumor MRI scan, and the image of the brain tumor MP MRI scan undergo the interpolation method for the image pre-processing, and after that we implement the deep learning models to predict the MGMT value in the brain tumor. So we use 2 deep learning models, ResNet-50 and the CNN models, to predict the MGMT value. The ResNet-50 give better accuracy, around 86% compared to CNN, which give only 85%. For future work, so we plan to get the real brain MRI scan in Malaysia to predict the MGMT value in the tumor. So the current project, we use the data set from the BRADS. It means the British brain imaging data set. So we want to implement in Malaysia for Malaysia database. And then we also plan to use the computer aided diagnostic to get the MGMT value in brain tumor. So we will develop the graphical user interface, the GUI, okay, to get the MGMT value for the brain tumors. And we also plan to apply other deep learning models to increase the accuracy in predicting the MGMT value. So these are the list of references. And that's all for my presentation. And thank you for your kind attention.